This last part of the lecture, we're going to be talking about how plasma membranes from two separate cells interact with each other, specifically how the plasma membrane from two different cells connect with one another through cell junctions. Now there are lots of different types of cell junctions, and you'll notice if you ever take a look in different biology textbooks, you'll even hear sometimes the same junctions being called different names, and it can be a little confusing. Your book, for example, makes a distinction between two types of junctions called adhesion junctions and desmosomes, and they don't, in my opinion, do a very clear job of describing the difference, and moreover, desmosomes is probably the term you're going to hear more frequently. So I'm going to have us focus on desmosomes and not really worry so much about the adhesion junctions at all. Okay, given that, there are lots of different ways in which cells can contact one another and anchor to one another. Sometimes cells will use what are called anchoring junctions to attach to one another, and the most common of these that you'll see are desmosomes. And desmosomes are like, you can kind of think of them as like spot welds. They're little points of attachment between two cells in various locations that allow the cells to still stretch. So if you imagine if I have one cell here and I have another cell here, and they have little plaque layers here with some little intercellular filaments between them connecting them, as you see in this picture here. If I were to pull on this cell in this direction and this cell in this direction, that spot weld would allow those cells to stretch but still remain connected. Okay, those are called desmosomes. Now compare those to tight junctions. In tight junctions, think of this like superglue. Here's an example of tight junctions. You have a lot of much smaller connections between the two cells. So if you had your one cell here and you had your other cell here, all of this would be a junction, which means if you pulled very hard in this direction and in this direction on this cell, the likelihood would be that the cells would tear apart from one another because there's no give in that junction or connection. The last major type of cell junction that you hear people talk about in animal cells are what are called gap junctions. And gap junctions are simply little channels or holes created by proteins that connect two cells together so that the fluid or cytoplasm in one cell over here can connect with the fluid or cytoplasm in a cell on the other side. And we'll learn in 202 that this is really a critical component for cells being able to communicate with one another rapidly. For example, it's because of gap junctions that all of the muscle cells in your heart can communicate with each other more or less simultaneously and thus contract as a single unit, even though it's made up of millions of individual cells. Now, outside of cells, there are a couple different things. One thing you see outside of all cells is something called interstitial fluid, and that's or intracellular fluid. And intercellular fluid is simply a fluid that bathes all cells. All cells are surrounded by fluid in the body. Also surrounding many cells in the body is this thing called the extracellular matrix, or ECM for short. And the extracellular matrix is made up primarily of lots of fibrous proteins, as well as some other things like proteoglycans, which is a fancy way of saying large molecules of sugar with a little bit of protein attached. Among the different proteins, there is a protein called collagen, like this guy right here, which is used to give the extracellular matrix surrounding the cells strength. And then there are smaller proteins called elastin proteins, which have an ability to be stretched and to recoil like a rubber band, and they give the tissue resilience, or the ability to recoil after they've been stretched. There's also involved out here in the extracellular matrix different types of adhesion proteins, and these adhesion proteins like fibronectins and laminins, we're really not going to get into too much detail right now. What you really need to know about these is that they allow the components of the extracellular matrix to communicate with the interior of the cell. So for example, if collagen fibers in the extracellular matrix begin to get ripped or torn, signals will be sent into the cell so that new collagen fibers can be made and exported out of the cell to replace the damaged ones. And we'll talk much more about the extracellular matrix when we get to learning about histology near the end of this semester, and also in Bio 201 at the beginning of next semester. Last thing I want to talk about very, very briefly is the difference between plants and animal cells. Of course, plant cells have a cell wall around them that the animal cells do not. So they have their plasma membrane, and then outside of the plasma membrane they have a cell wall made of cellulose. Now in fungi they have a cell wall too, but their cell wall is made of, if you remember, chitin. And in bacteria they also have a cell wall, but their cell wall is made of peptidoglycan. The presence of that cell wall makes it a little more difficult for two plant cells to communicate with each other, but plant cells do have something that's kind of like a gap junction called a plasmodesmata. And it's basically just a protein hole or opening that allows two cells to communicate with each other through their cell walls. And that wraps it up for the plasma membrane. See you in class.